Hello everyone. My name is Kevin A.D. Kernia, and I am a PhD candidate in the Department of Chemistry at Chung-Der Christian University, under the guidance of Professor Chung-Der Shao. In this video, I would like to introduce my recent publication titled Comparative Analysis of Zebrafish Fear Responses to 8 Different Fish Species Using 3D Locomotion Tracking Assays. In recent years, the use of zebrafish as an animal model has increased due to their high fecundity, low cost, and ease of genetic manipulation. Zebrafish are known to elicit behavioral responses, displaying anxiety-like, stress, and fear responses, shown by erratic movement, increased shoal cohesion, diving, and freezing behavior. As a result, zebrafish are a suitable animal for high-throughput behavioral study. In this study, zebrafish were exposed to 8 different non-predator fish species and their variations, and their behavioral phenotypes were observed. The relative body area size of the tested fishes ranged from 3.7 to 22.1 times that of the zebrafish, with the adult convict cichlid acting as a positive control for fear response and the tiger barb as a negative control. The fish that we tested are glass catfish, juvenile and fry convict cichlid, orange, red, and green-black tetra, clown loach, bala shark, Damascene cichlid, and threadfin acara. Zebrafish behavior was observed using a 3-dimensional recording setup to record the front view and top view of the tank. Zebrafish were acclimated to the tank for 5 minutes, followed by video recording for 10 minutes. The tested fish were then introduced, and the second recording was conducted to observe zebrafish behavioral changes. Zebrafish and the tested fish movement were then tracked using iTracker.ai tool, and 3D trajectories were reconstructed using the Fish 3D Locomotion Assay app. This is a more detailed preview of F3LA data analysis after zebrafish and tested fish movement were tracked using iTracker.ai tool and finally statistically challenged using GraphPad Prism 8. 8 major zebrafish behavioral endpoints were observed in this study, including locomotor activity endpoints, movement orientation endpoints, exploratory behavior endpoints, fractal dimension, entropy, shoaling behavior, and average distance to tested fishes. Locomotor activity endpoints were divided into 4 minor endpoints: average speed, freezing movement time ratio, swimming movement time ratio, and rapid movement time ratio. While movement orientation endpoints include average angular velocity and meandering, exploratory behavior endpoints encompass zebrafish vertical preference through time in top, middle, and bottom, the thigmotaxis, total distance traveled in top, and the number of entries to the top. In the locomotor activity endpoints, the zebrafish average speed was affected by the changes in swimming, freezing, and rapid movement duration. Reduction in zebrafish average speed was observed during exposure to tiger barb, glass catfish, juvenile and fry convict cichlid, and green-black tetra, while an increase in average speed was observed when exposed to Damascene cichlid. The reduction and increase in average swimming speed are mostly due to changes in rapid movement. Next, in the movement orientation endpoints, 2 minor endpoints were observed: average angular velocity and meandering, which usually represent erratic movement in zebrafish. From our test results, exposure to juvenile and fry convict cichlid, all colors of black tetra, and threadfin acara increased meandering behavior significantly. The third major endpoint is exploratory behavior endpoints. The minor endpoints observe zebrafish preferred vertical area, thigmotaxis, and top area exploration. From our test results, most zebrafish showed a reduction of thigmotaxis when exposed to test fish, except during exposure to threadfin acara. Additionally, zebrafish were observed to stay in the top area of the tank during exposure to all convict cichlids, all black tetras, Damascene cichlid, and threadfin acara. Zebrafish movement trajectories were also used to calculate the fractal dimension, entropy, and shoaling volume. The fractal dimension represents movement complexity, entropy represents movement predictability, while shoal volume represents shoal cohesion of zebrafish. Red-black tetra showed increased fractal dimension and decreased entropy, which means its movement is more complex and less predictable, while on contrary, exposure to bala shark and threadfin acara reduced zebrafish fractal dimension and increased zebrafish entropy. Additionally, zebrafish showed tighter shoaling during exposure to juvenile and fry convict cichlid, green-black tetra, and threadfin acara. Zebrafish and test fish trajectories were also used to calculate the distance from each zebrafish to the test fish. The results showed there is a significant difference between zebrafish response to glass catfish and convict cichlid. Zebrafish also showed shorter distances towards tiger barb in comparison to the convict cichlid, but statistically insignificant. The results showed zebrafish avoid adult and juvenile convict cichlid and demiscence cichlid the most. Zebrafish and test fish trajectories were projected to observe their movement. In the projected graphs, black lines represent zebrafish movement, while red lines represent the test fish movement. From these projections, we can observe zebrafish avoidance behavior towards the test fish, which is especially apparent when exposed to convict cichlid, demiscence cichlid, and threadfin acara. From this result, the zebrafish's top area preference might represent fear response. Then we conducted a principal cluster analysis and projected the heat map by combining all results obtained in this study. The analysis divided test fishes into two groups. First group contains control and our negative control, the tiger barb, along with glass catfish, orange and red-black tetra, and clown loach, while the other group contains our positive control, the convict cichlid along with its variations, green-black tetra, bala shark, demiscence cichlid, and lastly, threadfin acara. This result is quite similar to the time and top duration and zebrafish distance to tested fish's result, which means these two endpoints are quite strong to determine zebrafish behavioral response in this study. Due to the body size variety of the tested fish, we further test the relation between body area and zebrafish behavioral endpoints. Only the two most important endpoints are highlighted in this brief presentation: time and top duration, and average zebrafish tested fish distance. The correlation test showed no correlation between the tested endpoints and body area of all fish. But when the test was scaled down to include only the variation of the convict cichlid, there was a moderate correlation between time in top and body area, and a strong correlation between zebrafish tested fish distance and body area size. In conclusion, our study design exposed zebrafish directly to the tested fish for behavioral observation. According to our knowledge, this study design is the first to be reported, as previous studies often use images, videos, separators, or different containers to reduce mortality of zebrafish. However, this study uses non-predatory fish, therefore it was safe towards the zebrafish. Based on the results, zebrafish exposed to demiscence cichlid, threadfin acara, or green-black tetra showed similar responses to those exposed to the convict cichlid, which was used as a fear inducer in zebrafish previously. While exposure to bala shark seems to induce anxiety-like responses in zebrafish, however, this must be further tested in further study due to the unavailability of a positive control for anxiety-like responses.